All right, so we're going to go through the questions on quick quiz week 2 as quickly as we can, but more importantly, as accurately as we can, so you can identify the main ideas for each question that will be tested again on the exam, and then again at the end of the year in the end of your exam. Remember, you need to understand the concept. So even if you got an A or an B in on the quiz, if you missed an important idea, you need to make sure you understand that idea. Okay, so we see the hypothesis is designed to answer a question that was asked after making careful observations. So you make careful observations, you ask a question, and then you design a hypothesis to try to answer that question. The hypothesis has to be logical and it has to be testable. If it's not testable, you can't test it, it's not science. So when a scientist is ready to rest, and I meant test, okay? That's my mistake. When a scientist is ready to test a hypothesis, which of these might she do? Now, this is one of those ambiguous questions because one of your colleagues argued and made a good argument for after the hypothesis, we should go right to the experiment. My thinking when I wrote it was that a scientist, when they're ready to test a hypothesis, has to look at historical information, has to make additional observations from nature, and then do an experiment. Although this is correct, and this is that A, B, and C are correct, I can understand someone saying to themselves, well, we have the hypothesis, let's go right to the experiment. Let's design an experiment to test the hypothesis. My point with this question, again, I gave it to you just because I thought it was just maybe a little deep for the quiz, but it will be tested again on the exam. That after you get your hypothesis, you have to go and look and do research and see what other people have done to test that question, if it's been tested before. And so that you don't end up repeating other people's experiments, you want to make something new and you want to find out some new answers, not answers that have already been pretty well experimented with. You want to go back and look at nature and make sure that you are testing something that is that is in a way that it's possible. You might design an experiment that's impossible. You want to go back and make sure that your observations match up with your expectations and vice versa. So after you look at your historical information, you want to make sure that if you want to know that apples are falling from trees, every time something goes up, it comes down. You want to make sure that there are no examples in nature of something going back up and not coming down. So after you've done that, then you'd go ahead and design and do your experiment. So I went ahead and agreed with the student that made the argument that jumping to experiment C was I guess you could think of it as a valid answer, but it really, when you're talking about hypothesis, first you look at the historical information, then you make additional observations, then you experiment. Experiments are rigorous tests that determine if hypothesis is rejected or supported, never proven. And I understand that some people will say hypotheses are proven, the experiment proves it or disproves it. It's a use of weak language. The word proven is not appropriate. Some people would argue that it is, that some things are proven within a reasonable doubt. However, the problem with that thinking is that it locks you into this idea that, and it really will disappoint you, will end up disappointing you, that things have been proven or not proven. It suggests strongly, and in fact I can't even think of the word, it's a stronger word than suggests. It pretty much clearly states that if it's proven, then it's right, then we're ready to go. But nothing in science is proven. Nothing is unquestionable. So the thing that we say is a hypothesis is that we gather evidence to support it, or there's evidence that disproves it. Disproving we can do. It goes back to that question that I asked you in class. If I throw something up and it falls down, how many times do I have to throw it up to prove that it will always come down when I throw it up? And the answer to that question is an infinite number of times. You'd have to do it forever to prove that it would never keep going up. So the idea that the word prove is really kind of a misleading word that people do use in science, unfortunately, that really is something that really shouldn't be used in science. There are several types of experiments that can be done. Which of these will we focus on in this class? And this is just a question that I told you directly that this is the answer. I told you in class, I think it was Tuesday of last week, that it was the laboratory. We're going to be doing laboratory experiments. I described to you the different kinds of science that are done, different kinds of experiments that can be conducted. These are just 4 of them. But that we're going to focus on the laboratory experiments. Now, I described to you this experiment. I said 1 plate, here I had an antibiotic. I didn't use the word antibiotic. I called it substance X. Well, I did use the word antibiotic. There it is. But I called it substance X. I didn't want to say erythromycin or penicillin or anything. But I added antibiotic to A and not to B. And I want to know if these bacteria, these 6 types of bacteria, are going to be killed by this antibiotic. In other words, they won't grow on the plate. Or B, they do grow on the plate, whether they do or not. I grow them on B to make sure that that there's nothing wrong with my agar, that there's nothing in the medium outside of the antibiotic that's also killing them. Or maybe there's something wrong with the bacteria, that they just won't grow. So B is my control group and A is my experimental group. The independent variable is whether I add the antibiotic or not, and the dependent variable is whether the bacteria grow or not. So what is probably true of their experimental design? The scientists that did this, they kept everything but the use of substance X the same. Well, that's true. That should be something you should always do. You only change one thing in an experiment. So these are your 2 experiments. Between them, the control and the experimental, between these 2, you're only going to change one thing. And that's whether you use substance X or not. Everything else about them will be the same. The temperature at which you grow them, the humidity, the content of the agar, everything is going to be the same, except for that one thing, whether you added substance X or not. So A is true. A lot of people chose A. They chose A right away. But they didn't look at the other answers. So maybe B, C, or D are correct. So B, they repeated the experiment many times. We said in any good science experiment, I emphasize this over and over again, you must repeat the experiment more and more and more, as many times as you possibly can, in order to collect evidence that supports your hypothesis or even your theory. It only takes one time for this not to work and there not be some other thing that's impacting, confounding your results that would lead you to believe that this experiment's been disproven. So you have to do it over and over and over and over again in order to collect enough evidence to support your hypothesis so that other scientists say, well, it looks like this is the way it is, until somebody comes along and shows you that it's a different way. They will present the results at a peer conference. As I said to you before, all scientists have to present their results after they've achieved them without communicating their ideas, and that's the point here. This is the communication of their results at the peer conference. They must communicate their ideas, or their science is useless. And of course, the answer is all of the above. All 3 are correct. Now, someone asked, "How do I know? I can't read their minds. How do I know they're going to present at a peer conference?" Well, let's say that this is a questionable one. Let's say that she has a point, although I'll argue that most results are at some point or other presented at a peer conference. But let's go ahead and say that this is something that's up in question. In multiple choice questions, you always pick the best option. If you know A and B are true, and they are, without a doubt, and you have questions about C, D still has to be true. So your answer is clearly all of the above. It's the only one that could be. It's the only one that it could be. Okay. So 3 is all of the above. This 2 was A. 1 was again what I wanted was all of the above. But some people may have may have said, well, maybe it was just the experiment, right? So I would argue that that's not the best answer, that the best answer is, and I'm the one who chose chose the answer key. But because, as I said before, because I want to make sure that I don't take points away from you just because of an ambiguous question, I'll go ahead and give you a point there. All right. If the results are the same as expected, and substance X does not stop If the results are as expected and substance X does not stop bacterial growth on a plate A, and substance X does stop bacterial growth on plate A, when testing the 6 different species of bacteria, what is the next step? How generalizable are the results? So if I test 6 different species and A does stop the bacterial growth, what's the next step? If it works on these 6, it'll work on all type of bacteria. Well, I hope that everyone sees that how wrong that is, right? I hope that no one in here, no one in class looks at that and says, well, I think that that's reasonable. Well, that's not a reasonable comment. You can't possibly think that if it works on 6 bacteria, that it will work on all bacteria. Well, the next question be, if it works on plates, it will work on the human body. Just because bacteria, the antibiotic stops the growth on plate A, doesn't mean that bacteria growing in your blood will also stop growing, or that this antibiotic won't kill you if it's put into your bloodstream. So B is also not a good answer for this particular question. Since it worked on the experiment, further investigation should be conducted to see which type of bacteria it would work against. So the answer is C, and that's because once you have a positive result, you want to make sure that that bacteria is doing what it's supposed to be doing under the conditions that you want it to work. Okay, if you want it to work in the body, you got to make sure. That's where that whole clinical trials and all the other experiments have to come into play. So we said A and C above. Did I skip number 4? I guess I skipped number 4. When considering the experimental design, A is the experimental group and B is a control group. We said that's correct. A is correct. B is now A is the experimental group because in A we changed the independent variable. What was the independent variable? The presence of substance X. So A is where we changed it. B didn't have any independent variable, and that's why this B is a control group. All right, so this is your control. All right, so this is the control that should show. What this control should show is that there's going to grow. This should be your positive growth control, or if you want to say your negative control for your hypothesis, which your hypothesis is that the bacteria won't grow. So a positive for your experiment would be that the bacteria don't grow because you want to stop the growth of the bacteria. So right away you can get rid of B because it says B is the experimental group, and if you don't understand that B is the control group, you need to come and see me after school and let's talk about what the difference is between control group and an experimental group is. In 4, the independent variable is the use of substance X. As I said earlier in the video, that is the answer. And we used another experiment in the notes, but the idea is still the same. The thing that you're controlling is the independent variable. The thing that reacts or changes because of the independent variable, that's the dependent one. So the fact that whether bacteria grow or not, that is the dependent variable. So you're looking at C is correct as well. So A and C are both correct. So what you're looking at is letter D as the answer to that question. So let's go on to the next page. So a Mars mission 6 discovers in 2025, a Mars mission of 2025 discovers an object that seems to have order. All right, so it has order. So right away, I know some of you, again, jumping and said, well, it has order, and order is a characteristic of life, then it must be alive. But remember the virus example that you wrote an essay on. The virus has a couple characteristics of life, 1.5. If at our level of understanding of how a virus works, let's say it has 1.5 characteristics of life. But there's 6 characteristics of life. So you can't really just say, okay, so it has order, so it's alive. A computer has order. Do you call that alive? An engine has order. Do you call that alive? You open up your TV screen, your flat screen, you open it up. There's all kinds of order to it. We don't call it alive because it doesn't have the other characteristics of life. And that was the point of that discussion. So that's a big idea. So if you chose A because it has order and you identified the words here in the answer to the words in the paragraph, then you need to think about making sure that you're checking off all the boxes and then making an educated guess as to whether something is alive or not. Not automatically jumping at the idea that because it has one of the characteristics, we're done, it's alive. It has to have many of them, if not most of them, or all of them. So let's take a look at, so A we can get rid of. We can get rid of A. Let's go ahead and get rid of alive. It has alive. So because it's alive because it has order. If it had order and a bunch of the other characteristics, then you might argue it were alive. But it only has, so far it only has order. Say the B says not alive because it does not keep homeostasis. Well, that matches, doesn't it? Didn't we say that in class? If it keeps homeostasis, if homeostasis is something that an object has, then it's one of those characteristics of life. Let's read what the problem says about homeostasis. It says it does not seem to respond to any stimulus. So that's key for one of the other answers. It gets cold when it is cold in the environment. It gets warm when the environment is warm. So it's not keeping homeostasis. So one of the key factors in homeostasis is temperature, regulating temperature, as you'll see when we look at the human body a little deeper. So it can't possibly be not alive because it does not keep homeostasis. Not alive because I'm sorry. It cannot keep homeostasis. Excuse me. It cannot keep homeostasis. But then, so the question is not alive because it does not keep homeostasis. Well, that's correct, right? So this is. I know that there's not, and that's confusing. All right. So that's really difficult. All right. Maybe it could have been phrased better, but I have to say I've seen answers like this on exams. All right, so it says not alive because it does not, on standardized exams, not alive because it does not keep homeostasis. This object does not keep homeostasis. That is a characteristic of something that's not alive. So B is an answer. Is an answer. Does that automatically say I should circle B? Not necessarily. I have to look at all the other answers first. Alive because it does not keep homeostasis. Well, that one's wrong because we said if it's alive, it keeps homeostasis, not the opposite. So that one is wrong. So we could, even though this was one that people got confused about, this answer is the one that's wrong. Not alive because it does not respond to the environment. Now let's think about that. It says it does not seem to respond to any stimulus. Stimulus is the environment, so it doesn't respond. Now it does change volume when you change pressure, but so does an empty Coke can. So we really have to think about that. And if you're going to force me into deciding if something is alive, I'd have to say because of B and D, I'll say it's not alive. Does that mean that it's not alive? No. That just means that we, according to the information that we have, the best answer is E. Okay, excuse me. I can't get rid of this cold. All right, so when you go into the cold, you start to shiver. This is an example of which characteristic of life. Okay, in class, if you took good notes and you listened, I specifically used that 3 times as the example of keeping homeostasis. So really, A or C is the answer here. Homeostasis, the answer I was expecting. Now, some people argued, well, if you're shivering because of the cold, then you're responding to the environment. Well, they're right. This is why I gave you guys this question correct, because they're making a good point. And if you make a good argument, well, I have to listen. So here's homeostasis. This is the answer I expected. I told you that this was the answer I was expecting. I told you that this is the example of homeostasis. I must say that homeostasis, by definition, is also responding to the environment. So homeostasis is this idea that if it gets cold, you do something to warm up. If it gets warm, you do something to cool off. If your pH gets low, you do something to get your pH high. If your pH goes high, you want to do something to get your pH low again. It's about keeping that equilibrium, keeping it at that set point, like a thermometer, keeping it, your thermostat at home, keeping it at that set point no matter what happens. So this homeostasis is the answer, and unfortunately, I don't think I did a great job at trying to teach you that homeostasis requires responding to the environment. So this is one of those cases where homeostasis includes responding to the environment, but homeostasis is the best answer. Now, obtain and use energy, someone else, that same person argued that all the above was the answer, and that this is an example of responding to the environment. They also argued that you had to process, obtain, and use energy in order to shiver. Well, dang it, I can't argue it. So I went ahead and accepted all answers for this, and I said, you know what? But the answer, I guess, would have to be all of the above. My intent was, and I will rephrase it for the test, is that I want to know that you understand that homeostasis is this idea of balancing and keeping equilibrium. All right? So again, this is that characteristic of life question. What is the difference between growth and development and reproduction? Well, growth means getting bigger. Development means specialization. So you can think of a tadpole changing. Not only does a tadpole get bigger, some tadpoles don't get bigger, by the way, but not only does a tadpole get bigger, but then it also changes into a frog. So specialization or changes. So development means changing or specialization. Reproduction means making more copies. So when you reproduce something, you're making more copies of it, or you're doing something we'll learn later about mitosis. So when we're talking about these four, the answer here is D, letter D. All right. So a virus has many of the characteristics of life. Is a virus alive? Defend your answer using the characteristic of life. Write your answer in the back. Now that we'll talk about in the next video. That's the essay quiz. And if you see all these questions here, if you answered all these right, you should have easily been able to answer that. But there's issues with a few of the essays. Most people did really well. And by the way, most people are doing well. So you should all be proud of yourselves. But you got to get the details in order to get the high scores. Life is complicated, yes. Comes from other living things, true. Now see, again, when I saw mistakes, when I'm looking at answers there, people got it wrong. Often what people are doing is they're circling the first one because it's right. Well, it's right. And so is living things come from living things, and so is changing over time to adapt to natural selection, and so is passes information from one generation to the next. So the answer was all of the above. Now somebody told me that they thought I was trying to trick them because there's so many all of the abovs. You need to put your mind out of that. All you need to do is you either know your answers or you don't. If you know your answers, you're answering them. If you have questions about it, you have to use logic. Could there be tricks? Well, I guess some people do tricks. I'm not trying to trick you on a quiz. Life on Earth comes in many different forms. So many, in fact, that classifying life into 3 domains and various subdivisions becomes necessary to understand numerous fields of biology. In biology, this is an example of which unifying theme of biology. Well, it's the diversity of life. The diversity of life. And life is so complicated, and it's amazing. It's an amazing thing to study, but that is literally right off of the notes in class, and it shouldn't be that difficult to answer. What are these for? This is the idea that things change over time. Species change over time. Change. Structure and function are related. That's that door example that I gave you. Also the enzyme, the idea if an enzyme changes shape, it stops working. And the idea molecular basis of inheritance is just that DNA and RNA, specifically DNA, is how information is passed down from one generation to the next. Then there's cell theory, the idea that all life is the basic unit of life is a cell. So those are four of those big domains. One person in the 9th grade kind of confused this with the characteristics of life. This doesn't really the characteristic of life, as more of the fields under which people study, the things that we find that connect life together. So they're related, I guess, and you can find words that are similar in both. But this one is a little more detailed. All right, so enzymes, an enzyme's shape, and I should have put, there should be an apostrophe in here. An enzyme's shape is necessary for it to work. In fact, small changes in shape can impact everything from how a drug works to how well newly evolved character impacts survival. So which unifying theme of biology does this exemplify? I was hoping you would notice shape was necessary for it to work. Shape, or what we call structure and function. Structure and function. It's in the first word, structure and function. I was hoping you would choose B. That is the answer to this. All the others are incorrect. Although I know some people had arguments, they didn't hold water. So the answer is definitely B. Now they said, well, doesn't that also have to do with evolutionary change, they would argue. Well, evolutionary change is change from one generation to the next, or several generations within a species. So really, when we're talking about it being a small so when we're talking about shape is necessary for it to work, the only thing it could be a structure or function, not changing in species, not changing species over time. Molecular basis of inheritance. DNA doesn't change its shape, really. It's the sequence of DNA that changes. So it's still a double helix. There's still chromosomes, and in fact, the chromosomes are similar, if not exactly the same, between individuals. Well, not exactly, but the individuals have very similar chromosomes as far as number, size, and shape. So really, the molecular basis of inheritance only has to do with DNA itself. Evolutionary conservation is the idea that things stay the same between different species because they're essential. They're essential to how things work. So if those things change, then the thing breaks. The thing won't work. So the example I used is if you're going to redesign a car, you're probably not going to redesign the wheel, right? Because it's so simple and it works so well. Why would you want to redesign the wheel? You might improve on the wheel, but you would not make it square or triangular, because that just would make the car not work. So that's what happens with this idea of evolutionary conservation. If things are going to change over time, some things are going to stay the same because they're so essential. And finally, the diversity of life. And that we covered in question 11, and I think that it's obvious it's not that one. All right, so let's go to the next page. So here, there are important involved characters that are essential to how an organism works. These characters can be found in numerous species that are incredibly different and far removed. Now, isn't that what I described earlier about evolutionary conservation? So it's this idea that you have these things that are the same in a banana tree and us as human beings. So the banana tree has mitochondria. That is exactly the same, pretty much exactly the same enzymes and works exactly the same way as we have in our cells. But we're really different. I mean, no one's going to say that a human and a banana are the same or similar in any way. Not just that they're plants and animals, but their cells in a banana have the same as something that's similar to a cell from me. So that similarity, that mitochondria makes us similar in some way. That's a evolutionary conserved structure. That's something that's an organelle, obviously, but it's something that's been conserved over time. So it's something that we really it seems like this is a very important component of how things work. Sort of like if you were an alien species and you've never seen a car before, and you come down and you look at a car and you see that all the cars everything all that moves in the same way, all on the planet, everything has wheels, then you might think to yourself, as somebody from another planet that's never seen any kind of vehicle before other than a spaceship or something that hovers, you might say, well, whoa, this wheel thing must be very important. Well, that's what happens. That's what we do when we look at these evolutionarily conserved structures. We say, wow, these things must be important for how this organism works. So it's a really important concept, this evolutionary conservation. All right, so finally, evolution, how does it depend on? And this one, I think it's as simple as they come. I think if you got 14 wrong, we have to talk about why it's probably just a brain fart. Information being sent from one generation to the next. The next option was information being sent backwards. So you can't send your genes back to your grandfather. That's silly. That's not something I would believe anybody actually meant to say. Characteristics staying the same. Well, the whole point of evolution is that things are changing. Some things staying the same, you could argue there's something for that, but it's not really, it's not whether they stay the same or not. What it depends on, the best answer is information being sent down the line through time. Parent being the same as the offspring. If the parent's the same as the offspring, or the characteristics staying the same, then it's not evolution. Evolution is about change over time. But it does depend on information to be sent from one generation to the next. All right, so that was the quiz. I'll send you another one of these for concerning the essay. I hope this helps. Please review it. Have a great day. And prepare for next week's quiz. Get to know those organic molecules. They're going to be difficult if you don't study them. All right, bye.